Okay, good morning all. Good morning everyone. Good morning. Okay, good morning everyone. Okay, so friends, in the day before yesterday class, we have started the process of transcription, and if every one of you quickly recall, we have discussed exactly till this particular slide where we have given the overview of this particular transcription process, right? Good morning everyone. So exactly till this particular slide we started discussing it. Okay, and if every one of you quickly recall, friends, I have told you very clearly in case of a transcription or while synthesizing the RNA, we did not require entire strand of the DNA. We require only a small segment of the DNA only for the process of transcription. Good morning, everyone. Please continue. I hope friends, every one of you very clearly remember how many type of promoter, how many promoters are there in case of prokaryotic organism we studied. There are only two promoters are there. So one is the one is located at -10 and another one is located at the -35. Okay, then the next one, I hope friends all of you remember RNA polymerase enzyme. I want every one of you remember this thing without fail, friends. It is having two subunits, right? So friends, one we call it as the core enzyme subunit, and friends, another one we basically call it as the sigma subunit. So if all of you quickly recall, I told you that the core enzyme and sigma subunit do not have any active role in the process of transcription. If everyone quickly recall, what did I tell you? Sigma factor just help the core enzyme to recognize where the promoter is. That is the ultimate goal of this particular core sigma factor. It do not have any complete active role in the process of transcription. It will just help the core enzyme to recognize the promoter present on the DNA. So friends, now let's start about process of initiation, process of elongation, process of termination. Friends, the process of initiation will be, it's not complicated, it's little confusing. So I want every one of you pay utmost attention about this particular initiation. Okay, understand. Friends, the first step of the initiation we can call binding of RNA polymerase, closed complex formation, then friends, open complex formation. What is this closed complex and open complex? Friends, every one of you please kindly look into the diagram very carefully, friends. You all can see the diagram. This is our everyone pay attention. This is our RNA polymerase enzyme, and this is what factor, friends? Of course, this is the sigma factor. So friends, to this particular sigma factor, what it will do? It will help the RNA polymerase enzyme to come and associate with what, friends? To come and associate with the -10 region, as well as friends, to come and associate with the -35 region. And I want every one of you has to understand one very important point is that everyone pay attention. RNA polymerase enzyme with the help of sigma factor, first it is associated to with the region, which regions? -35 regions. So now the most important point you have to remember, friends, RNA polymerase associated with this particular promoters in such a way that it can form a very tight attachment. All of you listen friends, just imagine, friends, this is the promoter of -35. See here, RNA polymerase attaches very tightly. Okay, RNA polymerase attaches to the promoter region very tightly in such a way that you only try to look into this. Imagine my hand is the RNA polymerase. This entire area is the promoter. Imagine this is the promoter. It will attach very tightly, very tightly in such a way that RNA polymerase enzyme cannot move at all. That much tightly it attaches. How it can attaches that much tightly, friends? Of course, with the help of this particular sigma factor only. Friends, this is what exactly called as closed complex formation. Kindly do not forget this particular point. Now friends, now the question is very clear. Every one of you understand what I am talking. This particular RNA polymerase enzyme has to move from where to where? It has to move from